Hi, everyone. Thank you for joining us virtually today. I'd like to welcome our speaker today, Dr. Kelly Speer, who will be sharing her research on human and wildlife health through the lens of bat, vector, and microbiome interactions. Dr. Speer received a master's in science in zoology from the University of Florida and a PhD in comparative biology from the American Museum of Natural History, and is joining us today from Northern Arizona University, where she's an assistant professor. Her work explores the ecology and evolution of parasites and other symbionts through space and time, and relies on both field-collected and museum house specimens. I'm really excited to hear more about this work, and I'd like to invite you to join me in giving Dr. Speer a warm welcome.
Thanks very much. Thank you for the invitation. I'm super excited to be here today and to meet with a few of you afterwards. So let's just jump right into it. So I use a lot of natural history collections in my research, and I think natural history collections are a really underutilized resource when we go through the process of pathogen surveillance and pathogen surveillance for human health. And the reason why I think that is because natural history collections have this habit of continually reinventing themselves. If you imagine, for example, this specimen that was deposited over 50 years ago, or the specimens that were first deposited in collections in Europe hundreds of years ago, those collectors could not have imagined the ways that those specimens are being used today. For example, those initial specimens were collected before we knew what DNA was. And now, through the use of high-throughput sequencing like Illumina NovaSeq, we can generate billions and billions of reads of data, all to study these specimens that were collected so long ago, prior to our knowledge that this technology would even exist. We also have advanced in our ability to image these museum specimens through micro-CT scanning. So we're getting this really high-resolution 3D imagery of the internal biological organs and systems that make up all of these specimens and collections. So all of these components are new uses of existing specimens in museums. And recently, we're also starting to realize that museum specimens are not just representatives of, one individual of their species. They actually contain the entire suite of associated microorganisms and parasites, and we can learn about those microorganisms and parasites through these originally collected museum specimens. This is, for example, a fluorescent in situ hybridization image of the midgut of a museum fly. The red portions are fluorescing Bartonella. So we are using these existing specimens to learn more about their associated parasites and pathogens. So take, for example, this bat voucher, and this bat voucher is in a museum. And this bat voucher can tell you about the DNA, the RNA, and the morphology of the bat, but it also captures the associated ectoparasites, endoparasites, the microbiome and pathome, and environmental data. Together, this suite of information is known as the extended specimen. And through this extended specimen, we can use museum collections in conjunction with contemporary sampling to better understand the trajectory of evolution and ecology of host parasite and host pathogen interactions through time. Museums offer this unique snapshot of the past, so we can look at things like evolution of viruses and their hosts across time. Given we're starting to understand that organisms, the interactions of organisms and with each other and organisms with their environment are often complex reflections of their microbial associates as well. This type of extended specimen perspective is really invaluable for helping us address the big questions in biology, like what are the consequences of environmental change for biodiversity? What determines the susceptibility of a host to a pathogen? And how can we predict and prevent the next pandemic? So in my research, I'm an evolutionary ecologist. So I examine the interactions between bats, their arthropod vectors, and the microorganisms associated with both. This approach really lends itself to One Health efforts because it allows us to link information from the environment and from wildlife in holistic ways to address questions about how environmental change might lead to outbreaks of pathogens that are a concern for human health. So by taking this holistic perspective, we can learn more about the interdependent processes that determine those spillovers or those outbreak occurrences. So essentially the take-home message for this talk is that a community-wide perspective enables novel insights in wildlife and human health. So I'm going to take you through a few vignettes to demonstrate this perspective. The first, we're going to look at how does geographic distance influence biological communities. We know that geographic distance is an important factor when we're looking at the emergence of a novel pathogen. This is a map of the emergence and spread of white nose syndrome, a fungal disease of bats that's caused devastating declines in bat populations in North America. And it seems like the spread, the dispersal and occurrence of this pathogen is really determined by the migration and dispersal of the bat hosts. So geographic distance, understanding how geographic distance influences pathogen spread is really critical, and it's critical that we apply this community-wide perspective. The second vignette we're going to talk about is what can a community-wide perspective tell us about ecology and evolution? And the last one is how can we leverage this extended specimen idea in One Health? Okay, so just a little intro about the system before we jump into the data. Bats are ecosystem service providers. So they are really effective pest controllers. They eat their body weight in insects every night, and this can save farmers a lot of money in pesticide sprays, and actually makes communities where insect-eating bats occur safer because there are fewer pesticide sprays occurring in their area. Bats are also seed dispersers and pollinators of important crop plants like banana and durian. Bats are also infamous because they host many pathogens. And what makes them really unique is that their immune systems seem to allow them to tolerate these infections without showing disease in the same way that other mammals would. So they're model systems for understanding host-virus interactions. So I've circled a little arthropod on top of the head of this bat, and these arthropods are called bat flies. They're actually true flies, but they're true flies. They often look more like spiders, though. So bat flies are blood feeding. They're obligate blood feeding ectoparasites of bats. So they don't feed on anything other than bat blood, and they are vectors of bat pathogens. Including eukaryotic pathogens like a bat-specific malaria, viral pathogens, and bacterial pathogens. So there is some evidence that bats rely on gut bacteria to supplement their nutrition. If we think about the phyllostomid bats, a lot of them eat essentially only sugar water. They drink fruit juice all day, and that's all they eat. So how are they getting enough protein to subsist? How are they not getting diabetes? So there's some evidence that potentially the bacteria in their gut are provisioning nutrients that are missing in their diet. But this evidence is even much better supported for bat flies and all blood-feeding insects in general. Bat flies, like other blood-feeding insects, have primary endosymbiotic bacteria that provision B vitamins that are low prevalence in blood. So this reliance of bat flies on their associated beneficial bacteria is very strongly correlated. Okay, so we expect disease spillover events where a wildlife pathogen infects humans to occur more frequently as habitat is lost, native habitat is lost, or the separation between humans and wildlife decreases. So as we come into contact with wildlife, or as wildlife is stressed because native habitat is lost, those things all contribute to spillovers. However, we have almost no information about how this works for arthropod vectored pathogens of wildlife, despite its relevance, especially in North America and Latin America, where the pandemic potential pathogens are typically arthropod vectored. So this bat vector microbiome system allows us to test hypotheses about how environmental changes impact pathogen transmission in wildlife communities. It's a great system for doing it. Okay, so that's our setup. So, oh, sorry. Let me tell you a little bit more about bat flies. So there's two families of bat flies, the Nycterabiidae and the Streblidae. The Nycterabiidae have all lost their wings. They look a lot like spiders, and the Streblidae most of them have maintained their wings, so they do look like true flies. And they are closely related to two very interesting families, the Glossinidae, the tsetse flies, which are vectors of important human diseases in Africa, and the Hippobosidae, the head flies, which are vectors of agriculturally important pathogens. So by understanding how environmental change impacts the pathogen transmission of Streblidae and Nycterabiidae, we can start to develop hypotheses that might apply to these other two families that have human health relevance or agricultural relevance. Okay, so how do I collect these samples? So this is a very old video. Things have changed a lot since COVID of what it looks like at one of the field sites I visit in Belize. So all of those white bags on the line over there, each bag contains a single live bat, and everyone going up and tying flagging tape on that line is interested in studying some component of the bat's biology. So a lot of folks are working on bat flight because bats are able to fly and land in ways that could be relevant for rocket propulsion. Other folks are working on bat systematics. Other folks are working on disease ecology and parasitology. So we really make use of these specimens that we're working with. Okay. So I sample the ectoparasites and tissues from these bats. This is a very pregnant Pteronodus mesoamericanus here. And let me see if this video will play so you can see the flies running around on the wing. They're kind of difficult to catch. And so once we get them, we use a combination of sequencing technologies, including metabar coding, such as 16S rRNA amplicon sequencing to detect the bacteria associated with the flies and also the bat hosts. And we also use metagenomics, so a shotgun sequencing approach to get more loci, more information about those bacteria and associated pathogens and parasites. These are really low biomass samples. So if you are working on low biomass samples and you want to talk about it, I've spent a lot of time thinking about it, so I probably have some advice. Okay, so that's the setup. Let's get into our vignettes. So let's tackle this first one. How does geographic distance influence biological communities? So for this vignette, we're going to be talking about this bat. This is Leptonicterus yerba-buena, the lesser long-nosed bat. And in this photo, its face and head are completely covered in pollen. And that's because it is a pollen and nectar feeding bat. It is the primary pollinator of agave and cacti in Mexico. So if you like tequila, you like this bat. It is also a really useful bat for our question because it is migratory. So it spends the spring and summer in northern Mexico and the Baja Peninsula, and then the fall and winter in southern and central Mexico. This allows us to control for the impact of dispersal to examine the hypothesis that bat and bat fly associated microorganisms vary geographically or are highly conserved. And given that dispersal is high between these sites, it allows us to control for that variable. So here we're actually looking at the bat skin microbiome, not the bat gut microbiome. And the skin microbiome can influence susceptibility of bats to white nose syndrome, the fungal pathogen I mentioned earlier. So by looking at skin microbiome variation across these sites, we can start to understand that initial protective barrier to certain types of pathogens, especially the ones that first invade through the skin. Okay, so here are my sampling sites. We have a northern Baja site, 2 southern Baja sites, and 2 mainland sites. And we sampled 116 flies from 42 bats, and we took skin swabs from those bats. And then we also swabbed the 5 caves that we sampled them from so that we could test the hypothesis that bat skin and bat fly microbiomes vary geographically, or conversely, that they're conserved. Okay, so what did we find? Let's look qualitatively first. So this is just looking at relative abundance of different bacterial taxa from our different sample types. So here we have our cave on top with that trapezoid and the bats on the bottom. And each of those bars represents a different sampling site. Our Northern Baja site here, our Southern Baja sites here, our 2 sites, and then our 2 mainland sites here. And each of the colors represents a different bacterial taxon and then its relative abundance at that site in that sample. What you'll notice is, just off the bat qualitatively, is that there's a lot of inter-site variation. So the bat skin and cave microbiomes vary geographically, but interestingly, they kind of track each other. So the Northern Baja bat skin microbiomes look similar to the Northern Baja cave microbiomes, which is interesting. Let's take a look at our 2 bat fly species. So we have Trichobius sphaernotus and Nycterophilia coxata. Nycterophilia coxata is a laterally compressed ectoparasite, so it looks like it's been smashed in a book, and it's actually really difficult to get a dorsal picture. So this is actually from the side. This silhouette is from the side. Again, we have each bar representing a sampling site. And unlike what we saw with the bat skin and cave microbiomes, the bat fly microbiomes do not vary geographically very much. They are pretty conserved geographically, and they are very distinct from each other and from the cave and the bat skin. This is primarily driven by the abundance of their primary endosymbiotic bacteria. So here, Nycterophilia coxata has a genus-specific endosymbiont. It hasn't been named yet, so I just refer to it here as Nycterophilia endosymbiont. Whereas Trichobius sphaernotus is associated with Arsenophilus, which is a known endosymbiotic bacteria of bat flies. So those are very high in abundance and drive distinctions between the 2 bat flies, which are feeding on the same blood, the same bat individuals, and the bat skin is distinct, and the cave environments are distinct. 2 other bacteria that I want to point out to you, I've outlined in solid Bartonella and in dashed Wolbachia. So Bartonella and Wolbachia are much higher relative abundance in Trichobius sphaernotus compared to Nycterophilia coxata. Again, despite these flies feeding on the exact same bat individuals. So let's talk a little bit about those 2 bacteria. Bartonella is an arthropod vectored pathogen of mammals. Some members of this genus cause disease in humans. For example, Bartonella quintana causes trench fever. Although the human disease-causing species don't seem to be affiliated with bats or bat flies. Bat flies are the primary vectors of Bartonella in bats. Wolbachia, on the other hand, is a reproductive parasite of insects. So it leverages the reproductive system of insects to advance its own propagation. And it is sometimes a facultative symbiont. So it may sometimes provision vitamins or be protective to the ectoparasite from invading pathogens. So for example, some members of the genus Wolbachia provide protective capacity against malaria in mosquitoes. So mosquitoes infected with specific types of Wolbachia are actually resistant to malaria. So this is a really interesting bacteria to find in bat flies. Okay, so I showed you a qualitative look at what the microbiomes look like, but let's take a quantitative look. Are these actually different? So this is a principal coordinates plot. Showing our different sampling types again. Our caves are in this lighter yellow color, our bats are in this salmon color, and our bat flies are in purple and black. So they are distinct. the cave and bat skin microbiomes almost completely overlap with each other, but the two bat fly species are distinct from each other and also distinct from the cave and the bat skin. This is supported by Permanova, which is standardly used to look at significance in principal coordinates analysis of microbiome plots. But I should note that this data violates the assumptions of homoscedasticity that are required for Permanova. So in order to see if this data is still, if sample type was still a significant explainer for this data, we used random forest classification to develop a random forest model that classified our samples by sample type using their microbiome data. And the only time misclassifications happened was between bat skin and cave microbiomes. The bat flies were never mistaken for each other. So that's a good sign. It means that this data is very indicative that bat flies have distinct microbiomes from the bat skin and the caves. Okay, so bat skin and bat fly microbiomes variably reflect geography. There's strong geographic variation in bat skin and cave microbiomes despite high dispersal between these sites. Remember, these bats are migratory and occupy these sites annually. Bat fly microbiomes are distinct from their environment and from the bat skin. So this means that bat fly microbiomes are highly conserved, and this remained true even when we in silico removed their bacterial endosymbionts, which might suggest that even non-maternally inherited bacteria are strongly conserved in blood-feeding ectoparasites. And there's potentially differential susceptibility between bat fly species to Bartonella and Wolbachia, which is interesting depending on whether or not these are acting as parasites or facultative associates in these fly species. Okay, so that's how we can use this community-wide perspective to look at how geographic distance influences biological communities. So let's take a look at what a community-wide perspective can tell us about ecology and evolution more broadly, more fundamentally. So we know that environmental change is having significant impacts on communities. This is a map of forest loss through time. We have recent forest loss and historical forest loss overlapped with the human footprint. So areas of darker pink in these maps are higher forest loss and also higher human population density. So this forest loss is expected to result in the destabilization of communities. This can cause population declines, species extinctions, and it can increase pathogen prevalence by concentrating host species into small places. However, predicting the response of communities to environmental change is difficult, especially when there have been so few studies that take this kind of holistic perspective when examining community responses to environmental change. So here we can use this bat-fly microbiome assemblage to understand how environmental change might have cascading consequences for hosts, ectoparasites, and their microbiomes. We can use the island biogeography theory as a helpful null hypothesis in this instance. So according to island biogeography theory, larger source habitats indicated by this large gray circle can support more diverse communities and more individuals, whereas small, distant habitat fragments support less diverse communities and also fewer individuals. So this gives us our null hypothesis for how we might expect bat fly microbiomes to react to environmental change if they're adhering to expectations under island biogeography theory. So to examine this question, I worked with a Brazilian PhD student, Thiago Teixeira, and Thiago spent over a year in the field in the Atlantic forest of Brazil, sampling habitat patches of varying size and isolation, so that we could look at how it has these habitat patches have trickle-down effects for bats, bat flies, and microbiomes. So this large swath of green at the top of your screen is actually a protected area called regua. And Thiago sampled regua 3 times. And then all of these smaller green patches are remnant forest fragments surrounded by agricultural land that's been deforested. So the nice thing about this system is that these habitat patches have varying sizes and isolation from each other, so they fit well with the island biogeography theory. From these sites, we sampled these bat species that were the most common bat species across all sites. We have fruit-eating bats, blood-feeding bats, and insect-eating bats. And then we also sampled their associated bat flies. So we have some representatives from the Nycterobiidae and also the Streblidae. And in total, we had 222 bat flies from which we generated microbiome data. Okay, so what did we find? We found that bacterial richness in bat flies does not vary with habitat area or isolation. So in this plot, we have our sampling sites ordered by decreasing area. So we have our regua sites, which are the largest at this end, and our smallest sites down at this end. And then on the y-axis, we have our bacterial taxon richness. So how many bacterial taxa did we detect from each of these sites? And if this was following expectations of island biogeography theory, we would expect to see a very strong negative trend. So like high here, low here. We don't see that at all. Same thing with isolation. But the important thing to remember here is that bacterial richness is only one component of variation of the microbiome. Another very important component of variation, especially in bat flies which have conserved microbiomes, is changes in relative abundance. So in order to examine how environmental change was impacting relative abundance of bacteria at these sites, we used a type of ecological interaction network called a bacterial association network. So each of these, this is just an example from one of our sites. Each of these nodes represents a bacterial taxon that we detected at this site. The size of the node indicates the number of reads detected of that bacteria. And the nodes are connected with each other when two bacterial taxa had correlated changes in their relative abundance. So there can be a couple of things that can cause connections, correlated changes in relative abundance. One is that those bacteria could be interacting with each other, or it could mean that those bacteria are having synchronous responses to a host or environmental cue. So think about this, these networks as a way of visualizing those synchronous responses to a host or an environmental cue. So how does environmental change impact the relative abundance of bacteria, and are certain bacteria responding in correlated ways? Okay, so we see very similar networks for all of our regua sites. We have these isolated neighborhoods of interacting groups of bacteria called modules. And there don't seem to be many connections between those modules. So they're all kind of isolated. Then when we compare this to just 2 out of 10 of our fragments outside of regua, all of those 10 fragments have networks that look like this, where there are these long band-like networks instead of the modules of interacting bacteria. So they have these long band-like networks. So let's look at some summary statistics of these networks. We can see that modularity, so the degree that bacteria are interacting in neighborhoods of isolated bacteria, is much higher at the regua sites compared to the fragmented sites, which have a much broader range. So modularity generally measures network structure, and modules may be indicative of niches, although the research is still out on that. So highly modular networks, like the ones we saw in regua, the protected site, may be more robust than low modularity networks. And this is because low modularity networks, those band-like networks that we saw, if any of those connections is broken, it fragments the entire network. Whereas if a connection is broken in our modular networks, our regua networks, that only impacts the module, the neighborhood of bacteria, not the entire network. So the impact of change in the network is smaller. So those modular networks may be more robust. Another measure that we can look at is connectance. So connectance measures the number of realized connections compared to all possible connections in the network. So again, our regular sites show pretty low connectance relative to our fragmented sites. Connectance is may be associated with greater fragility, just like we saw with the modularity measure, where our fragmented sites may be more vulnerable to change. Okay, so what this indicates to me is that habitat fragmentation has cascading effects for communities. So the habitat patch determines which bat species are occurring there due to food availability, roost availability, etc. And then that host bat community determines the parasitic bat fly community present at those sites, which subsequently determines which associated microbiota might be there and in what relative abundances. But there may be this direct connection between habitat fragmentation and even ectoparasite-associated microbiomes through temperature. We know from Ixodes ticks that as temperature increases, the endosymbiotic bacteria associated with those ticks stops functioning, and the tick's microbiome enters dysbiosis. So if smaller habitat patches are also hotter, they could be directly impacting the relative abundance of bacteria in even ectoparasite microbiomes. So there could be this direct impact. Okay, so generally habitat loss seems to have these cascading consequences where parasite microbiomes reflect environmental change, which is kind of surprising considering parasite microbiomes are pretty conserved. The microbiomes may not follow null hypotheses like island biogeography theory established for animals and plants, which highlights a major gap in our ability to predict and understand how climate change might impact microorganismal biodiversity. And ecological networks offer a future route for examining these hierarchical communities and changing environments. Okay, so that was our second vignette. So let's talk about our last one. How can we leverage this extended specimen idea, this community-wide perspective in One Health? Okay, so I mentioned that I use a lot of museum specimens, and then I think they're an underutilized resource. So one of the ways that I think they're underutilized is in the study of viruses. So museum specimens haven't widely been used to study viruses. And this is largely due to their age and their preservation. So RNA viruses don't persist for very long, especially when museum specimens are stored at room temperature in ethanol without RNA in mind, essentially. Especially these museum specimens that can be 50, 100, 200 years old. But they can be extremely valuable for offering us opportunities to look at taxa we haven't looked at before. For example, this is looking at alpha coronavirus infection prevalence in 3 different bat species that hadn't been studied prior to this through rectal versus oral swabs. So which is going to be most effective for detecting these alpha coronaviruses in bats in the neotropics. It's variable by species, which swab type is better, but generally rectal swabs seem to be more effective than oral swabs, and prevalence is pretty high. And we wouldn't have known that alpha coronaviruses were in these species without looking for them. So one of the predominant ways that historical museum specimens are preserved is in formalin. And if we can find a way to get RNA out of those formalin samples, we can have a historical snapshot of what viruses might be like in bats 60 years ago. Okay, so I sought to do just that, to optimize an RNA extraction protocol for formalin-fixed museum specimens. And this isn't entirely out of left field. A lot of people have been working on very similar questions. So trying to get DNA out of formalin-fixed specimens, trying to get RNA out of human clinical samples that are formalin-fixed and paraffin embedded. And actually one group back in 2002 was able to get avian influenza RNA out of bird specimens, formalin-fixed bird specimens at the Smithsonian. So we used these combination of protocols to try and optimize one for bats and extracting RNA from bats. Okay, so generally speaking, when you're in the field, what happens is you collect a whole voucher specimen, and you can either put that whole voucher specimen in formalin and then transfer it to ethanol. The specimen remains at room temperature for its entire existence. Or before you put the voucher in ethanol, you can take a subsample of its organ tissues. And those organ tissues can either be stored in ethanol at -20°C typically, or flash frozen in nothing. There are some variations, but that's what we had available to us. So we had representatives that were only the voucher specimens in formalin. We had some that were the voucher specimens in formalin plus tissues in ethanol. And we had some that were voucher specimens in formalin plus flash frozen tissues. Okay, so then how do we get RNA out of formalin fixed tissue? So we tried a few different protocols. These are our 3 protocols that we tried. Essentially, the most important thing seemed to be effectively homogenizing the tissue. So then doing a proteinase K digestion to remove protein that attached itself to the nucleic acids, which is a known formalin causes protein to attach to nucleic acids. So doing this proteinase K digestion on well homogenized tissue gave us the best results. So what did the RNA look like that came out of this? Well, bad, as you might expect. So these were specimens that are about 35 years old, 30 to 40 years old. And so on the y-axis here, we have the RNA integrity number, and pristine, great samples typically have an RNA integrity number of higher than 7. And then on the x-axis, we have our RNA extraction concentration. So these red and blue dots down at this corner where we have low RNA integrity and also low concentration, these are the formalin fixed and ethanol frozen tissues. So interestingly, ethanol does not seem to be very effective at preserving RNA at -20, even though some folks still continue to use it. Our flash frozen tissues up here in green, those had a much better RNA integrity number, although not great. So that indicates that even flash frozen tissues, the RNA degrades over time, which we suspect would happen. So if we take a look at what this looks like, just in terms of a fragment analyzer. So here, this is kind of these seconds on the x-axis are kind of the length of RNA that we're getting, and the y-axis is fluorescence. So here are our 2 samples that were flash frozen. So we at least have a distribution of RNA lengths. For the rest of these, which were the ethanol frozen and formalin fixed, it's all just very fragmented RNA at the very far edge of our plot. So flash frozen samples, you at least have some distribution of lengths. Okay, so let's look at how proteinase K digestion impacted purity. So the 260-280 ratio is a measure of purity, and you generally want a 260-280 ratio of 2 for pure RNA. So our ethanol samples were very pure, the ones that never were in contact with formalin. Flash frozen samples, very pure. Formalin samples were a mix, even after proteinase K digestion. The lack of purity is likely due to remnant protein still attached to that RNA. So when we look at our extraction protocols, numbers 2 and 3 include a proteinase K digestion. Number 1 does not. So that proteinase K digestion really improved our RNA purity. So is this RNA usable for anything? Yes. So here's a qPCR using this RNA to amplify a mammal gene, a mammal housekeeping gene. So this is not viral RNA amplification, but this is mammal RNA amplification. So we saw no difference between our historical RNA and our modern bat RNA for amplifying via qPCR this housekeeping gene, which is great. So what about, doing a metatranscriptomics approach to see what we were actually picking up? So interestingly, most of what we detected was bacterial rRNA. And this is likely because bacterial rRNA is really heavily represented in GenBank in reference databases, but it could also be indicative that because bacteria often have cell walls, that their rRNA may be preserved better than the mammal RNA and the viral RNA in our samples. We did not detect, we did a panel for coronaviruses of bats, a general panel for coronaviruses of bats using qPCR. We didn't detect any in our RNA extractions, although our sample size was fairly small. We had about 50 bats from Wuhan, China, sampled about 40 years ago. Okay. So museum specimens are an overlooked source of RNA. Historical flash frozen specimens maintain medium quality RNA, but certainly sequenceable RNA. Even specimens preserved in ethanol or formalin fixed at room temperature maintain low quality RNA. So you can still get something out of there for qPCR. Museum specimens are valuable for examining this change through time in RNA viruses and in their hosts. I hope the take home here for you is that museum specimens offer we can get more information out of museum specimens than maybe we originally thought. Okay. So I hope I've convinced you that this community-wide perspective is enabling novel insights in wildlife and human health. And while we have always known that ecology and evolution of species does not occur in a vacuum, we can use new tools, new sequencing, new imaging tools derived from extended specimens to assess the ecology and evolution of species and risks to human and wildlife health in new ways. So taking this holistic perspective will be more powerful for preventing those spillovers or even the next pandemic. Thank you so much again for the invitation, and I'm happy to take any questions.
All right, if you'd like to ask a question, if you would raise your hand online, please.
Would it be better for me to stop sharing my screen?
Perhaps. All right, Megan, you have a question.
Yes, I have a question about the museum specimens and how you found them. So, I work with a lot of publicly available sequence data, and I think while I mostly access kind of the large databases, there's definitely these smaller repositories that people have and sometimes aren't maybe prompt about posting, and I think it can be challenging to track these down. Do you have any strategies when you are like looking for museum specimens or other collections that could be useful to you, and sort of how do you go about that process?
Yeah, so most museum collections have their data aggregated through GBIF, the Global Biodiversity Information Foundation, maybe is the F. So if you go to the GBIF website, you have to be careful because GBIF also aggregates from sources that include just if someone observed a species somewhere. So it's museum specimens like vouchers, tissues, but it's also just observances. Or observations. So go to GBIF and use the search for samples, and then you can choose specific museum collections that you want to look through, or you can set broader filters, and then that can highlight which collections have tissues that are relevant for the research you're interested in. And then each of those collections have publicly available databases on their websites, and you can get additional information on those databases. Another, Arctos is a very commonly used museum database collection management software. And if you go to the Arctos website, you can search all Arctos museum collections there also. Although those are primarily mammal collections, some bird collections, and some herb collections.
Thank you so much.
Yeah,
Right,
Elizabeth.
Hi, can you hear me okay?
Yeah.
Okay, great. Thank you. That was a really interesting talk. I really enjoyed it. I don't know anything about museums, or museum specimens, but I'm wondering if like your time kind of being in the field and now utilizing museum specimens, if you have feelings or thoughts about like future proofing specimens or like what people can be doing who work in museums to like make sure that specimens that are collected now are like as accessible as possible for stuff that we don't even know about that's coming in the future.
Oh yeah, this is the stuff that keeps me up at night. And a lot of museums are thinking exactly about this question. So there's a society, the abbreviation is Spinach, and they are a museum, a scientific society of museum folks thinking about these types of questions. And they had a symposium over the summer last year exactly focused on this, where it's specifically how are we going to collect specimens with the extended specimen in mind, and given we don't know the technology that's going to be invented and how specimens will be used in the future. The biggest challenge is, so people now do collect specimens in a broad variety of ways to account for all the possible things we can think of. So dry specimens, wet specimens, sampling organ tissue, flash freezing them before we fix the whole bodies, doing as many things as we know is, can help us. But the additional challenges posed by the extended specimen are difficult to think about because, for example, if we want museums to be able to help support surveillance of wildlife pathogens that could cause pandemics, we need to do a lot of sampling and a lot of non-lethal sampling. So how do we do that responsibly? How do we ensure that museums have enough capacity to store and manage those data sets? And how do we make sure those data sets are reaching epidemiologists and public health professionals and folks in biomedicine so that they can be used for the purposes for which they were collected? So yeah, I don't know if I have an answer, but those are all the kind of things that are like on the frontier for museums right now.
Yeah, thanks. Super interesting.
Yeah, David.
Hi, thank you so much for such an insightful talk. I feel like I've learned a lot about bats. I was just really curious, and this is, I've never worked with bats, vaguely, I just see them sometimes outside. But my question was related to Tiago Teixeira's work. I'm just very curious to know, like, how exactly the bats are sampled. Like, how do you guys catch them? Do you guys trap them? Do you guys like bait them? What does that method look like?
Yeah, for bats, it's we use a combination of nets. We're not able to bait bats very effectively. Unfortunately, it would be great if we could, because then we could get our sampling would be easier. But we use a combination of nets. The most common one is called a mist net, and it's also used to trap. It's also used to net birds. If you're familiar with how birds are netted, but it's essentially just a very fine microfiber net that essentially a fish net but on ground, spread between two poles, and the fibers are so thin that bats don't detect them until it's a little too late for them to avoid the net with their echolocation. So we set those. We can also use harp traps, which are set at the mouth of caves, and it's essentially two panels of clear filament strings. So the bats detect the first panel and go sideways to get through the first panel, but they don't detect the second one. So they hit the second one and fall into a bag where we grab them out. And then the last one that we typically use is called a hand net. So if we're going into a cave where bats are roosting during the day, we have essentially just a giant insect net that's on an extendable pole, and we put it up, the bats fall into the bag, and then we individually pull them out.
Gotcha, gotcha. Very interesting. Thank you so much.
So if we don't have any more questions, we'll go ahead and wrap up here. Thank you again, Dr. Speer, for joining us today. We'll take a quick break and then go into the breakout meetings. Thank you.
Great, thank you again.